Everybody's gotten settled in. We'll go ahead and get started. Good afternoon, good morning, and good evening to you, wherever you might be logging in from around the world. I am Rob Burgess. I head up business development for Sino-Biological, and it is my pleasure to welcome each and every one of you to the next installment in our BioTalk Tuesday series of live online webinars, where we hear about cutting-edge technologies as they apply to biomedical research, therapeutics development, vaccine development, and even diagnostic kit development. We have an excellent speaker and topic today focused on artificial intelligence as it applies to antibody-directed protein degradation. You're going to hear a lot about that today. Just one housekeeping issue that we have is I ask everybody that's on the call, first and foremost, to say hello to us in the chat box and tell us where you're from around the world. We're always very curious where people are logging in from. And also, if you would, please just type your questions for the speaker into the chat box, and I will run through as many of those questions as possible verbally at the end of the webinar today. And so with that, it's my pleasure, once again, an honor to introduce our speaker for today's BioTalk Tuesday conference, BioTalk Tuesday webinar, rather. The speaker is Dr. Miguel Moreno Garcia. Dr. Moreno Garcia is co-founder at Lumial Bio. So Dr. Garcia actually is a seasoned scientist with over 20 years experience in immunology and drug discovery across academia, as well as the biopharmaceutical industry. He earned his doctorate in cell biology from Sinvestov with training at the Trudeau Institute and the UCLA School of Medicine. He completed his postdoctoral fellowships both at the University of Washington and Seattle Children's Research Institute. Dr. Moreno Garcia has led discovery programs at Boehringer Ingelheim and Pfizer, advancing biotherapeutics programs which target immunomodulatory pathways in autoimmune diseases, as well as in oncology and fibrosis. He brings a very deep understanding and expertise in B-cell biology, translational biomarker strategy, and the development of innovative antibody-based therapies. And the title of Dr. Moreno Garcia's talk today is Artificial Intelligence for Antibody-Mediated Protein Degradation. What is possible today? And with that, Dr. Moreno Garcia, it's great to have you today, and I will turn the presentation over to you.
Okay, thank you so much, Rob. Can you hear me okay?
Yeah, you sound great. Thanks.
Perfect. Okay, thanks for the introduction. And first of all, I'd like to thank the Xenobiological team for the invitation to give this talk in the BioTalk seminar series. And yeah, as the introduction said from Rob, my name is Miguel Moreno Garcia. I'm the co-founder of Lumial Bio, and today I'm going to give an overview of what is actually possible to do with AI, especially for developing antibodies that help in the process of protein degradation. And I want to highlight that this talk, the intention is to give an overview mainly to biologists, people that would like to learn more about what they can do to actually boost their process of therapeutic development using antibodies, more like to preach into the computational biology, which actually are the experts. So this is more like for people that is in the lab doing their experiments and thinking about what can I do, what models or tools can I use actually that employ AI that can help me develop my therapeutics. And with that, I like to move on now to next slide. Let's say it's not moving. There you go. This is Lumial Bio. We are two co-founders, Lucrecia Marquez Rosado and myself. And we have, as Rob mentioned, in the intro more than 20 years of experience combined in academia and in industry in multiple areas of research, including cell biology, medicine, cancer biology, immunology. And we have had cross-functional senior leadership roles at large biopharma like Boehringer, Ingelheim, Pfizer, as well as Nurix. And we decided to found this company, Lumial Bio, with the mission to actually advance new type of therapeutic solutions uh, by developing a novel class of antibodies that not only are designed to bind and block the target, but also aiding in the degradation of the disease-associated targets. And this way, we are convinced that that would effectively address the limitations of conventional monoclonal antibody therapies. And I'm going to start actually with that part. Well, first here with the outline uh, of the talk. First, I will talk about why we believe that degrading extracellular proteins is way better, more effective than just blocking molecules. Then I will go into describing an overview of some of the antibody modalities that many companies are developing in these areas, focusing on one specific modality that is those that lead into the lysosomal compartment or degradation. Then I'll jump into where AI can actually assist on developing these type of modalities. And I'll show a few case studies based on these modalities from a couple of companies. And starting with the limitations of antibody therapies, there are several limitations in addition to advantages, of course, uh, that may limit actually the efficacy of these antibodies. One of them is actually the target occupancy. So it could be very incomplete. So if you dose a patient, those antibodies would bind to the target, but many of those targets may stay free. And because the targets are not modified at all, these targets may continue with the pathology process. Because of that, patients would need larger and often frequent dosing of the antibodies to actually cover and give a therapeutic effect, but also that can lead to strong and significant adverse events, side effects as well. That's one of the limitations. Another one is depending on the target. Let's say targets are multimeric, dimeric, trimeric, let's say like TNF alpha, for example. The antibodies against it can bind because they bind the same epitope. They can bind multiple TNF alphas, and then in that way cause a process of aggregation, multimerization of the antibodies. And that can be very detrimental to the patients. It can induce immune activation, complement activation, hypersensitivities, but also in terms of PK effects, also that can lead to very rapid clearance and elimination of the antibodies. And if they accumulate in tissues and cluster in tissues, that can also lead to tissue damage, for example, in the kidneys. Another disadvantage or limitation is that, especially for surface receptors or ligands expressed in the plasma membrane, when these antibodies bind to these targets, by the fact that they can cross-link these molecules and cluster them, they can induce unwanted cell activation processes, like induction of cytokine storms, for example, and severe systemic immune reactions, which can be very detrimental for patients. And also the fact that they cross-link these receptors or ligands, they can be quickly internalized in the cell and then degraded in the lysosomes. So losing the effect of the antibodies over time and then needing more doses for patients. So because of some of these limitations, we are convinced that we need an additional strategy. And one of them is actually using the antibodies to lead towards protein degradation or target degradation. because the key concept here is that degradation focuses mainly on reducing the functional protein levels, not just blocking their activities. And one of the reasons here, and I already mentioned that, is the occupancy can be very insufficient for highly abundant, redundant, or targets that are shed in circulation. And the strategy of using antibodies is also very attractive because the antibodies give them specificity and selectivity to your target of interest that is highly expressed in your disease of interest. And the main goal here is not just blocking the target, as I mentioned, but remove completely and inactivate the protein pool that will lead to a greater level of efficacy. Some of the mechanistic advantages that we're convinced extracellular protein degradation brings to the table using antibodies, one of them is removing the signaling capacity. And this is obvious. You eliminate the target. It could be a receptor, it could be a growth factor, a cytokine, and then you completely remove the signaling pathways from these targets. Another is with these modalities, you can bypass very high turnover or redundancies from these targets. For example, there's a target that with a small molecule, you block the kinase motif. So the molecule doesn't have any longer any kinase activity, but many of these molecules we know have adaptor functions and motifs that can continue with some signaling processes. By inducing protein degradation, you forget about it. Just eliminate completely the protein, and then you bypass that redundancy. Um, Another aspect which is very important as well with protein degradation is that your dosing can be substoichiometric, meaning that you don't need a 1-to-1 ratio, so one antibody for one target. So you can have one antibody, and that antibody may be able to induce the degradation of multiple targets at the same time. And also co-opting receptors that can recycle. And I will give some examples in the next slides about modalities that use receptors that help internalize the complexes into lysosomes and then recycling those receptors back into the surface and then releasing again the antibodies to the extracellular medium to continue with the process of capturing more targets and eliminating more proteins that way. So that's one of the advantages also of thinking about protein degradation instead of just blocking. And of course, these can expand to multiple targets, extracellular targets, and also difficult receptors that cannot be targeted or blocked easily with small molecules, for example. So now I'm going to be talking about giving an overview of antibody modalities that many companies have been developing recently that co-opt this idea of using protein degradation with antibodies. And I'm going to focus mainly on modalities that actually lead the extracellular proteins into the lysosomes in the cell for degradation. One of them is the LITAC. Many of you probably know very well about this. This mechanism uses lysosomal receptors, and the champions of these, on one side is Lysia Therapeutics, but also Eli Lilly has been working on this. And the main concept here with the LITACs is that, let's see, and those molecules are attacked with those glycans, then they are captured by these receptors, they internalize and then degrade in the lysosomes. So these companies now are co-opting this biology and then creating now antibodies that on one side bind to the target of interest or the protein of interest, mostly soluble molecules extracellularly. And then in the Fc portion of the molecules, they put these specific glycan modifications. And in that way, you have now a therapeutic that captures and harvests the targets floating around in the tissues, and then they bind to these shortening receptors through these glycans, and then they get into the endosomal lysosomal compartments for processing and degradation. That's one modality. Another one similar, where Genentech has been one of the pioneers, is the APTAC or PROTAPs, in which you have now a bispecific molecule or antibody, in which one arm of the antibody binds to the target, the protein of interest, and the other arm binds to an E3 ligase, for example, ring finger 43 or zinc finger 3 as well. And the idea is that by causing this clustering between the target and the E3 ligase, now the E3 ligase is in proximity to ubiquinate the protein of interest and then internalize the protein and degrade it in the lysosomes. Another modality that is similar to the APTAC, but now is using chemokines and chemokine receptors, are the kinetacs, I think, or kinetacs. I don't know how do you say that, the right pronunciation of that word. But the main concept here is exploring the fact that many chemokine receptors actually turn over very fast in the cell, like CXCR7, for example. And what these companies are doing is they are making fusion proteins where they have a region and an arm that is an antibody-like arm that binds to the protein of interest, and on the other side, they have the fusion of the fused chemokine like CXCL12, for example. And then by doing that, now they bring together the chemokine and the target. Now that induces internalization very quickly so that it goes into the lysosomes for degradation. And finally, one that is classical, very well known, is engineering the FC portion of the antibodies so that now these FC domains of the antibodies can bind with specific affinities or binding potencies to FC receptors, for example, FC gamma RTB. And by doing that, so you capture your target, and then these antibodies have very high affinity for these receptors that causes internalization right away, and then degradation in the lysosomes. Roche, Chugai, Regeneron have been working on these type of modalities. So these, in a nutshell, some of these modalities. There are many, many more. So I just want to focus on these four. And I'm going to give some case studies with the LITACs and with the APTACs in the next slides. So now an overview. Where do AI, or artificial intelligence, is currently being used actually to develop this type of molecules, or what we can do with AI in this case? So first, selection of your epitopes, your targets, by creating 3D structures of both the antigen of interest as well as your antibody, and then docking them, finding which of the regions in the antigen would be the best regions actually to target with an antibody. And there are multiple models that are AI-based and that you can also use using generative AI, like AlphaFold 3, ESMFold, IgFold, CollabDoc, for example, that can help you create these 3D models and then docking them. So you can now find different poses and then select those that better fit to the antigen with less clashes and liabilities. Another one where AI can assist is also on predicting affinities, changes in affinities, increasing the affinity of your antibody by modifying the paratopes, the CDRs. And AI can help proposing different CDR edits to give a more mature antibody, and also can help on ranking the different epitopes in your antigen and the paratopes in your antibody, so that you now move on with the antibodies that have the properties that you want in terms of binding affinities. And there are multiple models that you can use. BOLS2 is one of that, is very popular. MindFilter, for example. Also, AI can assist a lot in the design of multispecific geometries and linkers. For example, helping us define arm placements and target distances and angles. and then filling linker sequences and interface residues, as well as knocking to holes for bispecific molecules for the actually correct chain pairing. And here, for example, diffusion models have been very popular as well. And finally, in terms of developability and PK parameters, it is well known that computational biology is very important to predict with AI sequence liabilities, humanize your antibodies so that they are less immunogenic, also predicting aggregation liabilities, and also engineer the FC portion so that the antibodies are more stable and less prone to aggregation. And there are multiple models that you can use there listed here as well. So this, in a nutshell, very highlights where AI can support with these models. That many of them are based on deep learning, machine learning. So some of the gates that can be used to successful produce or eliminate extracellular proteins by degradation. Here, AI can assist very well by quickly identifying failures at each of these gates and eliminating those models that may not fit very well into your concept, and helping you actually before you do very costly wet lab cycles. One of these gates is on the biology, asking the question, do my modality would be able to remove the target in relevant cells and tissues, where potential issues you can have is that your receptor is absent in that tissue, or the receptor is shed, for example, or the molecules are shed, or you have very weak internalization, misaligned trafficking. So AI can help selecting receptors and different antibody formats that may fit actually the expression context of your disease of interest or your cell of interest. Also focus on epitopes. So saying, if you have this target, so you can define what is the right position in that target that your antibody needs to go to actually boost the process of binding, internalization, and degradation. Another gate is on the geometry of your antibody. So whether the complexes of antigen antibody binding are they can assemble physically. They can have a productive orientation. So because we know we can have multiple clashes, state clashes, incorrect spacing and angles, glycan blockages, extracellular molecules, proteins are usually highly glycosylated. So you need to be sure that your antibodies are not directed towards areas that are highly glycosylated, and also areas that are not too proximal, or the motifs or the epitopes are not proximal to the or facing the membrane, so that the membrane doesn't occlude the binding of the antibody to the target. So, and in this case, AI can help us create and prioritize different poses of binding between the antibody and the targets, doing initial assessment of steric clashes. If your format is not the most ideal, it can investigate different linkers and different formats to help you choose the best format for your therapeutic concept, to meet the distances and orientation requirements of the antibodies to actually target the molecule that you are interested in for the process of internalization and degradation. And finally, and very important is on the drug, whether your therapeutic is going to be actually developable. We know that we can have multiple issues with antibody development, like aggregation, issues with viscosity, stability, thermal stability, that can affect heavily on the PK/PD profiles later on. So AI can help us design screenings, developability screenings to flag specific liabilities, to prioritize, and that will help us prioritize candidates that will be able to survive the different CMC and PK parameters. So now going into the case studies. As I mentioned, I'm going to show you about LITACs and APTACs, where AI can support us here with the LITACs. First of all, and I already mentioned that it would help us building the 3D models of the antibody and the antigen. If you don't have the crystal structures, you can get these high efficient models, very similar models, where you can now pose these antibodies with the ligands in this, sorry, with the targets or the antigens, and judge the best epitopes where the antibody can bind um, by doing multiple poses and rank these different poses and say, this is the best pose, the best binding in this specific area of the antigen that I should follow on. The other aspect is, especially for this type of modality, is that AI can assist a lot on predict unstable capture by modifying the affinity and avidity of the antibodies. And this is very important because with this modality, this system, the antibodies need to bind with high affinity to the target so that it doesn't detach, floating around, before actually it binds to the shuttle receptor. So here AI can assist on creating antibodies with sufficient affinity so that it ensures that the antibody will be stuck to the antibody and then be able to bind to the shuttle receptor for internalization. And another aspect that AI can help using bioinformatics is identifying or picking what is the right shuttle receptor that you should pick. This is going to depend on your cell of interest, your disease of interest, of course. And also on the analysis of the antibodies, it can look at the FC portion and find areas that can be exposed that are not related to the paratope or cannot block the paratope, and also areas that would protect the binding to FcRn, for example, so that you don't affect the half-life of the antibody. So here is also helping with the ternary structure. Now you have, the binding of your antibody to your target of interest. You want to know if the antibody would bind to the shuttle receptor. So you can form these ternary complexes using like bolts2, for example, alpha fold 3, and making sure that any of the poses that you select actually fit also and binds to the shuttle receptor of interest. And AI can help defining that also with these models. And importantly, and I think this is a little bit more tricky, is refining the endosomal pH behavior. AI can predict the different behaviors of the binding of the target with the antibody and the stability of those bindings based on the protonation, just to ensure that once it gets internalized, the interaction of the antibody with the target is not released too early, so that now it's spit out, but also not too late, so that the antibody goes also into the lysosomes and gets degraded as well. So you need to find kind of the sweet spot of acidity or acidification in the endosomes to know that your antibody is going to be released, and then the target is going to go into the lysosomes, and the antibody is going to recycle back into the surface to capture more antigens or more targets. So AI can predict if the antibody antigen would stay bound through the endosomes and would raise the confidence that the cargo is processed in the lysosomes. The cargo in this meanings is the target. And of course, the in silico PK evaluation, just to anticipate any clearance trade-offs before actually you go into synthesize your antibody, and also all these developability triages that I already mentioned to start dropping down or dropping out any liabilities early before you generate your antibodies. So of course, AI can help mitigate some of the potential pitfalls, and it's very important to think about the pitfalls early on. For example, one of them is that the epitope can be occluded by the members on glycans. I already mentioned that. So that AI with these models can help you find better facing spots on the protein, in this case on the target, and flag areas in the target that may be covered by sugars. And then eliminating those areas or not, and selecting the ones that avoid all these potential liabilities. Another one is that for the shortening receptors, right? The endogenous ligands or glycans may be very abundant. So AI can help on estimating receptor crowding and also suggest stronger multivalent designs for the glycans that you can put on the antibody so that they outcompete the endogenous ligands. But if that's very hard to do, then AI can suggest differential depending on the expression of those molecules in the disease of interest or the cells of interest to you. Another pitfall is that the handles, in this case the glycans, those can break binding to FcRn or cause aggregation. So with AI, you can highlight regions that you say you do not touch these regions and all these sticky patches and check if the tag location is stable and safe. The pH behavior is also critical, as I mentioned before. So you want your antibody to fall apart too early, but also not too late. So AI can assist here by predicting how tightly the complex holds in the endosomal at the endosomal pH, and suggest tweaks in the paratopes in the CDRs so to change the sensitivities up and down. And there are multiple models that you can use for that. And of course, the PK sync, the pitfall is that may be stronger than expected. So AI can help you early on run what if scenarios. They are just predictable. They are not going to tell you exactly what's going to happen, but these scenarios can help you say this pose or this interaction here maybe lead me to a PK sync very quickly. So these scenarios can help you define which of those interactions may not be able to actually do that, and then you can move on with those. So another case study is the APTAC, very similar to the LITAC. For example, with the APTAC, you can build the structures of your arms, your antibody arms, for each of the two sides. In one side is the arm that binds to the protein of interest or the target, and the other side it binds to the E3 ligase, the ring finger 43, for example. You can create those models. You can build also the 3D structures of your protein of interest as well as your the E3 ligase, and then create these ternary complex and poses, mainly using the extracellular domains more than the whole protein. And AI can also help in refining what is the E3 ligase that you need to move on. So it can help you using the well-known bioinformatic tools in public databases. What is the expression levels of the E3 ligases, surface E3 ligases in multiple tissues and cells and in diseases. And in that way, you can select what is the E3 ligase that you need to use with your protein of interest. Very important here, especially for bispecific antibodies and multispecific antibodies, is the geometry that is going to be productive, in this case, to induce the ubiquination of your target of interest. So these angles and distances are really key, and AI can help you define those angles and distances by defining, these epitopes. You should consider these epitopes instead of these epitopes, these other epitopes, because these other epitopes may be very close to the membrane, and then your antibody won't be able to reach that, and they won't be able to cause the interaction between the two molecules at the same time. And this is very important for this type of bispecific modalities. And importantly, AI can balance the affinities of the two arms binding to your targets of interests. It's important here for the bispecific antibodies to bind with equivalent affinities to both the E3 ligase and the protein of interest, so that it doesn't bind preferentially to one or to the other. And then you cause a sink and you lose efficacy because it's binding preferentially to one arm. So here, AI can help you adjust the different affinities of the protein of the antibody against the protein of interest and against the E3 ligase, so that when the antibody binds the molecules, they remain engaged. And similar to the LITACs, it supports, it can help us in modifying the pH behaviors of the interaction between the antibody and the protein of interest and the E3 ligase as well, so that it goes into the lysosomes for degradation. And the developability and TMDD PK analysis to keep the constructs manufacturable, anticipating exposure and dosing risks from target mediated clearance. So in some of the mitigations also in this area, how AI can assist mitigate those potential pitfalls. One of them is that the E3 is not co-expressed with the protein of interest, and you need to know that before actually you create your therapeutic concept. So you can, with the use of AI, you can read the expression of multiple E3 ligases in your tissue of interest, in your cells of interest, and points towards the E3 that is actually expressed in the cell that you're interested in targeting, and the tissue and in the disease. So this is also very important. So your target of interest must have points of ubiquination. Of course, intracellularly, AI can help you scan the protein layout and say, intracellularly, you have motifs that contain lysines, and these lysines can be easily ubiquinated by the E3 ligase. If you don't see that in your protein of interest, maybe that target is not the right target to develop this type of modality. And AI can help you also eliminate or confirm that your protein of interest is actually the right one. Then also the geometry for bispecific is critical, where the two antibodies, one of the pitfalls is that the antibodies they can bind to different arms with different affinities, or that the antibodies may be mispaired, dimerizations, heterodimerizations to produce a proper bispecific. So an AI can mitigate this by trying multiple poses, checking these assemblies, then for the mispairing, suggesting linker lengths. For example, if you use, instead of an IgG with knobs and holes, maybe single chain FPs with a linker, suggesting linker lengths, for example, arm positions, to help you identify actually both arms would be able to bind to your targets of interest and to the E3 ligase. So another pitfall is the mispairing of the chairs and FC issues. So very similar to what I mentioned before, is checking assembly in 3D, flagging these mispairings and proposing interface fixes. So, and with this, I like to mention that many of the models that I just mentioned here, you can use each of them individually. You can get them and then get licensed for them, but also you can use web apps that have all these models, and you can have a subscription to these web apps. One of them that the company is using is NeuroSnap. This is a very user-friendly web app that had all these models, or many of the models that I discussed today in the webinar. And this is very friendly. This is not very friendly because you don't need to know any coding to actually create your pipelines. And it has access to many of these. And here what I'm just highlighting is at the bottom some of the models that I mentioned in this webinar that are in this NeuroSnap app, web app. So, and with this, let me see what is the timing. This is the conclusion, the takeaway, how we can use AI. First, it can produce very reliable 3D structure models that you can use them to reason about the epitopes, the geometry of your antibodies, and choose your best designs. Then make these designs computable to propose and run concrete designs in silico actually before you go and clone your molecules. For these specific modalities of intracellular protein degradation is in the lysosomes, is the risk subcellular routing in silico by predicting whether the complex could potentially reach the lysosomes. And importantly, front loads the development and PK checks by screening any potential risk in terms of manufacturability as well as exposure early on before actually going silico you go and produce your antibodies. Doesn't replace experiments. So you need to, it only helps you triage and reduce instead of, screening thousands of combinations. Now you narrow down to maybe dozens or at most hundreds of molecules. And that eliminates all these dead ends that will allow you to do a very quick experiments and answering mainly your more critical questions, accelerating your drug discovery process, development, and the launch. And with this, I like to finish. So we have time for a few questions. Thank you so
Great.
much for your attention.
Thank you, Miguel. That was a fantastic presentation. We really appreciate it. Very well done. Lots of information there. I will go ahead and start with the questions that we have from our audience. And I ask again for everybody who's still online to please type in some good questions into the chat box so we can ask them of our speaker today. The first question today is from Ahmed Solomon. Ahmed asks, I think, a good question here. Ahmed is asking, what about targeting a phagocytic receptor that naturally recycles and avoids lysosomal degradation? Like, how would you do that? Do you have any thoughts on that?
So, meaning that the receptor will never reach lysosomes.
I would assume that's what Ahmed means. So it naturally recycles and finds a way to actually avoid the lysosome and avoid degradation.
Yeah, I think here, as I mentioned, there AI can help on changing the pH behavior of the binding of the antibody to the target,
Mhm.
and it the ideal there would be to ask, for example, the AI models whether you can increase the affinity of the, let's say, the sugars and the ability, maybe more than the affinity, the ability of the sugars towards the receptors, right? The glycans that you put on the FC portion, say, I want these glycans to be of high avidity or low avidity there so that when it gets into the cells, it doesn't show back down, up. So if you, let's say, if you put these abilities too strong, then it may keep stuck to the receptor and then they go up. But if the ability is not that strong, then once you get a specific pH, maybe you can modulate that as well. Then it releases into the endosomes, and then the complexes go into the lysosomes.
Mhm.
It's a trick. I would say that's tricky,
Mhm.
where you need to do a lot of combinations. Um, of glycans and also modifying the paratopes potentially also to modify that interaction.
Mhm.
Uh, but it's very important to know the biology of your receptors, right? So that you know that they're going to be able to reach a site on the endosomes that once they release the target, it goes into degradation, not goes back into the surface.
Right.
Yeah,
It does seem like a pH optimization strategy would be the way to go in that respect. So
exactly.
great question, Ahmed. I'm asking others to please type in some questions. And while we're waiting on those, I can ask you, and I think you alluded to this maybe in one of your last slides about others having access to AI-based technologies for optimizing, their own research and eliminating a lot of the bench work that they may need to do. Do you license out your AI technology, or is it free to use, or what's your business model regarding partnerships or just others using your technology?
So specifically for, you mean for a company?
Yeah, yeah.
Uh,
For your company specifically, Lumial Bio, how do
yes,
you or
yes.
do you allow full access of your AI-based technologies to the research world?
Right. No, this is a very important question. So right now we are in the process of optimizing our platform,
Mhm.
and the platform would be able to define the best antibodies that we want to produce. And for us as a company, next step is actually go ahead and produce these antibodies and do the proof of concept for ourselves.
Mhm.
So we are early on. in the development of our therapeutics with our company with that specific concept. And the idea later on, of course, once the business progresses, is to as a potential exit strategy would be to develop therapeutics and go with companies for acquisitions. But also, we don't discard the possibility of licensing this technology once it's up and running and
Mhm.
efficient, I would say. So
Mhm.
that's one of the ideas. No, that's a good question. Thank you, Rob.
Great.
Yeah.
Yeah, no, it's a great business model. I think it's very standard and normal. You want to optimize the technology before you consider outsourcing it or allowing access by external parties, and you have to work out the compensation model as well. I fully
Yes.
understand that. And maybe you mentioned it in your talk, but can you say a little bit about the early drug concepts that you have without breaching anything sensitive? Any particular areas of disease or physiology you're focused on, and any exciting preclinical results yet?
Well, so at this point, I won't be able to mention exactly what we are trying to develop.
Mhm.
But the main idea is in the mission statement that I put there is that we want to develop antibodies that bind to the targets and aid into the process of protein degradation.
Mhm.
So I cannot disclose more than that, unfortunately. But once everything is in place in terms of IP security and all that stuff, we would be able to. But yes.
Yeah, I don't blame you one bit. I knew I was reaching for straws, but I thought I would try. So
All right.
no worries at
Okay.
all. I think we'll wrap it up with that. Miguel, again, I want to
Okay.
thank you. That was a fantastic presentation you gave. I also want to thank all of the attendees. We had a great attendance today, folks from as far away as Shenzhen, China, as well as Sweden and Mexico, and a variety of individuals from across the United States and North America. I also want to thank my colleague, Ms. Kayla Sarmiento, for executing another flawless symposium, Biotalk Tuesday webinar. And before I sign off here, I just want to mention our self-signaling breakthrough symposium to be held on Saturday, April 25th in Seattle at the Renaissance Hotel. And at this symposium, we will have the world's leading cell signaling experts talking about signal transduction, both internally within the cell and cell to cell communication, and how it applies to really human physiology and disease and the design of therapeutics and vaccines. And it's just going to be a fascinating 1 day seminar and webinar, I'm sorry, 1 day conference in Seattle, and I recommend everybody consider attending that. Whoever logged in today will get an invite for this symposium at the close of this webinar. And so with that, I will close. Miguel, thanks you once again for an excellent talk, and Kayla, thanks for your efforts, and thanks to the attendees who attended. And so with that,
Thank
we'll
you.
we'll
Thank you.
sign off. Thank you.
My
Very
pleasure. Very nice meeting you, Rob and Kayla.
nice meeting you. Thanks.
Bye-bye.
Bye.